Hello dear students, we are going to discuss today on micromeritics. The basic objective of present lecture is to make students aware of the fundamentals of micromeritics. The lecture also gives detailed description of particle properties like particle size, particle shape and particle surface area. Such properties are used for characterization of individual particles, particle size distribution and powders. What is micromeritics? Micromeritics is a science and technology of small particles. The unit of particle size used is micrometer, micron and is equal to 10⁻⁻⁶ meter. The study involves the characterization of individual particles, particle size distribution and powders. Particles are characterized by particles like size, volume, density, surface area, porosity, flow and other associated properties. The application of micromeritics in pharmacies are releases and dissolution. Particle size and surface area influence the release of the drug from a dosage form. Higher surface area allow intimate contact of the drug with the dissolution fluids in vivo and increases the drug solubility and dissolution. Absorption and drug action. Particle size and surface area influence the drug absorption and subsequently the therapeutic action. Higher the dissolution, faster the absorption, hence quicker and greater the drug action. Physical stability. The particle size in a formulation influences the physical stability of the suspensions and emulsions. Smaller the size of the particle, better the physical stability of the dosage form. Dose uniformity. Good flow properties of granules and powders are important in the manufacturing of tablets and capsules. Now we will see particle size and size distribution. Knowledge and control of the size and the size range of particles are of significant importance in pharmacy because the size and surface area of a particle related to the physical, chemical and pharmacological property of a drug. The particle size of a drug can affect its release from the dosage forms that are administered orally, parenterally, rectally and topically. In the area of tablet and capsule manufacture, control of the particle size is essential in achieving the necessary flow properties and proper mixing of granules and powders. Size of the particle is readily expressed in terms of its diameters as follows. The surface diameter, that is ds, is the diameter of the sphere having the same surface area as the particle. The volume diameter, that is dv, is the diameter of the sphere having the same volume as the particle. The projected diameter, that is dp, is the projected diameter of the sphere having the same observed area as the particle. The Stokes diameter, represented as dst, is the diameter which describes an equivalent sphere undergoing sedimentation at the same rate as the asymmetric particle. The sieve diameter, that is ds, is the diameter of the sphere that passes through the same sieve aperture as the asymmetric particle. The volume surface diameter, that is dvs, is the diameter of the sphere that has same volume to surface area ratio as the asymmetrical particle. From the above description, it is possible to conclude that same particle possesses different values for the diameter. Appropriate diameter should be selected depending on the intended purpose for which it is being applied in pharmacy. Particle number. The number of particles present in a dosage form should remain constant in order to maintain dose uniformity. This is important when drug particles are mixed with other ingredients to prepare tablets and capsules. Larger the number of drug particles, smaller is the error. Errors are large when the number of drug particles is small. Particle number is important in case of low dose or potent drugs. The number of particles per unit weight, n, is expressed in terms of volume number mean diameter, that is dvn. Assuming that particles are spares, the relationship for n is on your screen. Looking towards the particle size distribution, any collection of particles is usually polydisperse. It is therefore necessary to know only the size of the certain particle, but also how many particles of the same size exist in the sample. Thus, we need an estimate of the size range present and the number of weight fraction of each particle size. This is the particle size distribution, and from it we can calculate an average particle size for the sample. When the number or weight of particles lying within a certain size range is plotted against the size range or mean particle size, a so-called frequency distribution curve is obtained. This is important because it is possible to have two samples with the same average diameter but different distributions, frequency distribution curve. In general, the normal distribution curve is symmetrical, that is the bell shape around the mean. In this type of distribution, the positive and the negative deviation from the mean are uniform. Normal distribution is not found in pharmaceutical powders because of uneven size reduction processes. The distribution of particles in a powder is termed as unsymmetrical. Seeing the log normal distribution curve, in this type, frequency on y-axis is plotted against log mean particle size on x-axis. Advantage of this curve is that distribution pattern is made symmetrical when compared to normal distribution curve. Powders obtained by crystallization and milling methods exhibit log normal distribution. Second curve is cumulative frequency distribution curve. In this plot, cumulative percentage oversize or undersize is plotted against particle size. Sigmoidal curve is obtained. The advantage of this plot is that one can directly read the percentage within any given size range without any difficulty. Log probability plot is drawn by taking log particle size on x-axis and cumulative percentage frequency of probability scale on y-axis. In this curve, cumulative curve is converted into a straight line. Now, we will see different methods for determining particle size. Many methods are available for determining particle size, such as optical microscopy, sieving, sedimentation, and particle volume measurement. First one is the optical microscopy, which ranges from 0.2 to 100 micrometer, sieving, which ranges from 50 to 1500 micrometer, sedimentation, that is 1 to approximately 200 micrometer, particle volume measurement ranges from 0.5 to 500 micrometer. A guide to range of particle size applicable to each method is on your screen. Optical microscopy, which ranges from 0.2 to 100 micrometer. The microscope eyepiece is fitted with a micropeter by which the size of the particles may be estimated. According to the optical microscopic method, an emulsion or suspension is mounted on the ruled side of the mechanical stage. The microscope eyepiece is fitted with a micrometer by which the size of the particle can be estimated. The ordinary microscope used for the measurement the particle size in the range of 0.2 to about 100 micrometer. Some practical considerations: the microscopic method of measurement should be carefully standardized, otherwise considerable errors may be introduced. The sources of errors include the choice of diameter, technique of slide preparation and sampling. The value of projected diameter depends on the orientation of particles on slide. The diameter obtained in this method is actually the diameter of the circle whose area is equivalent to the projected area of the particle. The diameter is estimated using a graticule which is used in the microscope eyepiece. A less tedious method is double image microscope. In this technique, two identical images are observed in the field of view. A beam splitting device that is present between the objective and the eyepiece moves the image apart. The amount of displacement is indicated on the scale. When the images are edge-to-edge contact, the diameter can be read from the electric meter in the Trimble instrument. Advantages of these microscopic methods include: the microscopy allows the observer to view the particle, agglomeration or particles and any contamination in the powder can be detected. Particles in the dispersion must be free from motion. This can be avoided by mounting the sample with a cover slip. easy and simple. There are some of the disadvantages with the method, which are: the diameter is obtained from only 2 dimensions of the particles. The number of particles that must be counted, that is 300 to 500, to obtain a good estimation of the distribution, makes the method somewhat slow and tedious. Other methods are: popular measurements are projected area diameter, Martin diameter and Ferret diameter. Projected area diameter, it is a diameter of a circle with the same area as that of the particle observed to the surface on which the particle rests. It corresponds to number 3 in figure. Martin diameter, it is the length of the line that bisects the particle image. A line may be observed in any direction but must be drawn in the same direction for other particles measured. The Martin diameter is represented by number 1 in the figure. Ferret diameter, it is the distance between 2 tangents on opposite side of the particle parallel to some fixed direction. It corresponds to number 2 in the figure. The second method, which is the sieving method, which ranges from 50 to 1500 micrometer. Standard size sieves are available to cover a wide range of size. These sieves are designed to sit in a stack so that material falls through smaller and smaller meshes until it reaches a mesh which is too fine for it to pass through. The stack of sieve is mechanically shaken to promote the passage of the solids. The fraction of the material between the pair of sieve size is determined by weighing the residue on each sieve. The result achieved will depend upon the duration of the agitation and the manner of agitation. Practical consideration in the sieving process: care should be taken in order to get reproducible results. The type of motion influences sieving. Vibratory motion is most efficient, followed by side tap motion, bottom pat motion, rotatory motion with a tap, and finally rotatory motion. The type of motion of the shaker is fixed and standardized. Shakers are commercially available. Other factors are weight of the sample and duration of shaking. Sieves are produced by photoetching and electroforming techniques, are used to get a better estimate of size distribution analysis with the lower limit of estimation of particle diameter, that is 5 micrometer. Advantage of sieving is it is inexpensive, simple, rapid with reproductive results. The disadvantages includes lower limit of particle size, that is 50 micrometer. If powder is not dry, aperture become clogged with particles, leading to improper sieving. During shaking, attrition occurs, causing size reduction of particles. This leads to error in estimation. The third one is the sedimentation, and the range is 1 to 200 micrometers. In this method, sedimentation of particles may be evaluated by measuring the terminal settling velocity of particles through a liquid medium in a gravitational centrifugal environment using Anderson apparatus. This method can be used in formulation and evaluation of suspensions, formulation and evaluation of emulsions, determination of molecular weight of polymers. The rate of settling of particle in suspension or emulsion may be obtained by Stokes' law, and it is shown on your screen, where V is the rate of settling of the particles, H is the distance of fall in time t, rho s is the density of particle, rho 0 is the density of the dispersion medium, g is the acceleration due to gravity, and rho 0 is the velocity of the medium. Practical consideration includes the suspension should be dilute, which is up to 1 to 2%. For the estimation, powder is disposed in the medium to get a suspension. Sometimes particles form aggregates or get clumped together and fall due to its weight. This leads to error. A proper deflocculating agent has to be added to keep particles free and separated so that each particle sediments individually. Therefore, a dilute suspension is preferred. Flow should be laminar, that is, rate of sedimentation of particles must not be too rapid to create turbulence. Any turbulence flow will affect the sedimentation of other particles, thereby causing error in the estimation. The type of flow, whether turbulent or laminar, is indicated by dimensionless Reynolds number, that is given by Re, which is V d rho 0 upon rho 0. If Reynolds number is greater than 0.2, the flow is turbulent. Sedimentation method predicts the exact particle size. If Reynolds number does not exceed 0.2, the Anderson pipette apparatus consists of cylindrical vessel of 550 ml containing a 10 ml pipette sealed to a ground glass stopper. When the pipette is placed in the cylinder, the lower tip is 20 cm below the surface of suspension. The procedure is as follows. Prepare 1 or 2% suspension of powder in a suitable medium. A deflocculating agent will help in uniform dispersion of the suspension. Transfer the suspension into the Anderson vessel. Place the stopper and shake the vessel to distribute the suspension uniformly. Remove the stopper and place the two-way pipette and securely suspend the vessel in a constant temperature water bath. At different time intervals, 10 ml samples are withdrawn using two-way stopcock and collected in a watch glass. Samples are evaporated and weighed. The weight or the amount of particles obtained in each time interval is referred to as weight under size. The weight are converted into cumulative weight under size. Particle diameter is calculated from Stokes' law, with h in the equation being the height of the liquid above the lower end of the pipette at the time of withdrawing the samples. Particle volume measurement ranges from 0.5 to 500 micrometer. In this type of machine, the powder is suspended in a electrolyte solution. This suspension is then made to flow through a short insulated capillary section between two electrodes, and the resistance of the system is measured. When a particle passes through the capillary, there is a momentary peak in the resistance. The amplitude of the peak is proportional to the particle size. Counting is done by the computer. Going towards the particle shape is related to geometric shape and surface regularity. Particle shape will influence the surface area, flow of particles, packing and compaction properties of the particles. It is possible to determine whether the shape of the particle is spherical or asymmetrical. Discussing about the particle shape is related to geometric shape and surface regularity. Particle shape will influence the surface area, flow of particles, packing and compaction properties of the particles. It is possible to determine whether the shape of a particle is spherical or asymmetrical. A sphere has minimum surface area per unit volume. Therefore, these properties can be compared for spheres and asymmetric particles in order to decide the shape. The shape factor of particle can be expressed as ratio of surface to volume factors. Shape factor is equal to alpha S upon alpha V, where alpha S is a surface area factor given alpha S pi ds square upon dp square. Alpha V is volume factor and is given as pi ds cube upon 6 dp cube. The minimum possible value for shape factor is 6, which represents a sphere. If the ratio exceeds this factor 6, the particle is considered as asymmetric. Seeing the particle surface area of particle in powder can be calculated using particle size data, which can be obtained by any one of the methods discussed above. Specific surface area can be defined as surface area per unit volume given as SV or per unit weight given as SW. Taking the general case for asymmetric particles where the characteristic dimension is not yet defined, SV is equal to surface area of particle upon volume of the particle, which is given by n alpha S d square upon n alpha V, where d cube is as upon alpha Vd. Since alpha S and alpha V is equal to 6, SV is equal to 6 upon d. SW is equal to surface area upon weight is equal to n alpha SD square Vs upon n. Alpha d cube Vs rho, where rho is the density, is equal to alpha S upon alpha V into d Bsp. that equals to 6 upon dVsP. The above method is based on particle diameter data. This method can estimate the specific surface accurately for non-porous solids. Normally, porous materials have high specific areas. These materials have irregular surfaces and contain pores and crevices. Hence, the above method cannot account for these pores. Therefore, it is necessary to estimate specific surface area of powder directly. An accurate and direct method is necessary owing to its importance in release and absorption of drugs. The commonly used methods are absorption and air permeability. Now, going towards the conclusion, micromeritics is the science and technology of small particles. It has its applications in drug release and dissolution studies. Stability, dose uniformity and drug action. Properties like particle size, particle shape and particle surface area are studied in micromeritics. Such properties define the overall powder characteristics.